Alrighty, so we're going to kind of continue on this path of um, talking about the formation and evolution of species. So in the previous couple lectures, we were talking about how we identify species, the different mechanisms or concepts that we use, as well as how we kind of look or express different species relationships through the use of phylogenetic trees. Again, we're going to continue down this path and move into how you have the formation of species, how that process actually occurs. As per usual, I have to give a disclaimer. If you hear disgusting kind of slimy, chewy sounds in the background, that would be Figaro. I had to give him his moist food because he was climbing all over my computer and started sending emails. So I apologize if that causes a disturbance in the background, and if there are any interruptions due to squeaky toy attacks. So let's keep our fingers and toes crossed that none of that happens, but let's not hold our breath as well. So let's talk about how species are actually formed. How do we get different species? When we talk about the evolution of species and kind of the changes that occur within or between species, there's two major pathways that we address. There's kind of two major evolutionary pathways for changes in species. The first pathway is called microevolution, which we'll talk about in another video. Microevolution refers to evolutionary change that occurs within a species or a small group of organisms. And those periods of change occur usually only over a short period of time. But the more important fact is that these are changes within a species group. And so these are changes that can allow for kind of subspecies or small variations or kind of subtle changes within that species grouping. The other pathway, which we'll focus on for this lecture, is macroevolution. Macro meaning large. So macroevolution is referring to these long-term, large-scale evolutionary changes that occur either at or above the species level. So this is where you see the development of major patterns or changes among living organisms over extended periods of time. Now, you might be wondering what are some events or some examples that could represent these macroevolutionary events. Well, some examples include the multiplication of species or the diversification of species. Right? Those are both large-scale changes that occur first at and then involve an above approach at the of the species level. Right? You have the actual creation of new species, and they usually take long, extended periods of time to occur. Other examples would be the origin of novel traits or morphological structures. Right? So this could be the origin of kind of advanced snakes or venom delivery structures in different snake species. It could also be the origin of feathers. Another example of a macroevolutionary event would be kind of the mass extinctions that then lead to or are followed by these kind of explosive radiative events in different species that are able to kind of adapt to the environmental changes from that extinction and exploit those niches that are now open and available. Right, a great example of this would be the radiative event of mammals and the major diversification of mammal species following the CT extinction event. Mammals were able to occupy those niches that were once occupied by the now extinct dinosaurs, and that allowed for this major diversification and radiative event. So when we talk about macroevolution, there's kind of two sources of evidence. People always want to know, how do we know that this actually occurred? Is this hypothesized or is this real? Well, this is fact. Most of this we know for certain because we have evidence of it. Most of the evidence comes from comparisons between living organisms, and a great deal comes from the fossil record. The fossil record provides evidence of these major biological changes and kind of evolutionary events throughout kind of the history of life on Earth. Now, macroevolution itself, or the macroevolution of a species itself, happens as a result of speciation. Speciation is the evolution of new species by either a genetic change or a change in form. So you can kind of think of it as the branching off of individuals from the species in which they were originally from, right? It's a divergence event. Now, when that population branches off from the original population, it will go through the course of natural selection, right? Natural selection will occur, and as that happens, these individuals that make up this new group may build up adaptations that are no longer compatible with species, or sorry, with others of that original species. And if they're separated long enough, you can get large enough differences accumulating in the genetic sequences, or they're no longer able to interbreed. They then become different species, right? So this group that originally branched off has now become a different species that's distinct from the original species. So once this group separates from the original species, it'll develop its own unique characteristics based on the selective pressures it experiences or the demands of that environment, or kind of the operation of natural selection. But whatever the case, those characteristics will differentiate this new grouping from the original, from their ancestors. Now, the major concept to remember and kind of continuously reiterate to yourself is this concept of reproductive isolation. In order for a new species to arise, to truly have definitive new species, some members of that original species must become reproductively isolated from the rest of the species. This means that they can no longer interbreed or mate with members of that original species group, right? So once they branch off, in order for that branched group to become a true new species, you cannot have the individuals of that new group mating with the individuals of the original, right? That's reproductive isolation. And there's various ways that this can happen, that you can have kind of a reach or attaining reproductive isolation. We're going to talk about uh, 4 kind of major types or modes of speciation. I have them broken out or kind of grouped based on how reproductive isolation occurs. So reproductive isolation will occur because of some barrier. Some barrier prevents the free flow of genetic material between the populations of interest. And that's how they become reproductively isolated. Now, for some, this is a physical barrier. For others, it's not physical. It can be something that causes kind of these major changes in genetics, or it could be kind of this gradual or graded change in characteristics. When we talk about kind of the modes of speciation that involve physical barriers, these would be allopatric and peripatric speciation. So in both of these cases, geographical isolation occurs first due to the presence of a physical barrier, and because of that, you attain reproductive isolation. The other two, parapatric and sympatric, that barrier, again, you still have a barrier that is formed, but it's not physical. So a non-physical barrier enables reproductive isolation to occur. So we're going to go through each one. We'll go more in depth with allopatric, parapatric, and sympatric, but I'll give you kind of a quick overview before we kind of continue on with this lecture. Allopatric speciation in general refers to kind of the creation of new species because the original population was separated by an actual physical barrier. And that barrier would get in between members of the population and therefore separate them. Peripatric speciation, you can think of as kind of this extreme case of allopatric speciation. It's more of this extreme case of isolation where you have some event that causes kind of the isolation of individuals at the periphery of the original population. And over time, they will form 2 distinct populations, or they will obtain reproductive isolation. For your sympatric speciation, this refers to, or this will occur in populations where you see that the individuals are not separated at all. The individuals that are going through reproductive isolation are within that original population. They're not physically separated. So they all live in the quote-unquote same place. The other similar type of speciation that I had grouped with sympatric is parapatric speciation. Parapatric speciation, kind of in this mode, again, populations are not isolated by a physical barrier. Instead, they're kind of situated side by side with each other. So there's nothing, there's kind of this incomplete barrier or reproductive barrier here. There's nothing truly stopping the individuals in the population from mixing and mating. So usually kind of the product is these two distinct species with a hybrid zone held in between them, because you can have the mixing of some individuals from this population because they are held or situated next to each other with that incomplete barrier. And when you have two individuals from two different species mate, you get hybrids. So when we talk about hybrids and hybrid zones, that's a product of parapatric speciation. Right? So those are, again, the major 4 modes of speciation. We're going to talk about kind of the big 3: allopatric, parapatric, and sympatric. With allopatric speciation, this again involves that physical separation of populations, kind of starting from the tippy top. When we have a normal population, kind of looking at the original population that we're starting with, that population is what we would call geographically continuous. Right? There's no barrier that has yet formed or separated the population, and it has normal gene flow or normal patterns of gene flow, meaning that the movement of alleles across that range of species is relatively freeform, because the individuals can move and then mate with the individuals throughout that population. Each one has an equal opportunity of mating with an individual of the opposite sex. So you have this kind of equilibrial flow of genetic material throughout the population. You have a relatively homogeneous gene pool where each allele is represented in that population. Now, at some point in time, you'll begin to have this gradual formation of a geographic barrier or physical barrier that can come to isolate parts of the population. Either it'll isolate, it can break the population down in half, it can break them into smaller individual populations, maybe it kind of separates the individuals that are held at the extremes of the species range. Whatever the kind of the product of that original physical barrier, you have the separation of populations, and these populations now become geographically discontinuous, meaning that original free flow of allelic material is now prevented. It's hindered, or may not occur at all, because those populations are isolated by that physical barrier. Now, when separation continues for an extended period of time, the two populations, or more than two populations, are able to evolve along different trajectories. Their allelic frequencies at numerous genetic loci will gradually become more and more different as new alleles independently arise by mutation in each population. So each population is going through different mutagenic events or changes in physical morphological structure based on the selective pressures that they're experiencing. And the changes that are evolving in each population are independent of one another. Right? So the changes that are happening in population A are going to be independent and different from the changes that may be occurring in population B, for example. So once you have kind of, again, that separation, and then this divergence in evolutionary trajectory of those populations, that's allopatric speciation. Again, involves initially that physical barrier. Now, what can serve as a physical barrier? Well, that's a pretty easy question. So these barriers can be geographic, climatic, anthropogenic. You can think of examples or agents that are like continental drift or climate change, even changes in sea level. Any formation of a physical object or development in the landform that physically separates those populations and isolate them from one another. That's what we mean by these physical or geographical barriers. Right? So it could be maybe the formation of a new river or a river delta in a new area that kind of floods out. It could be the erosion or the process of erosion that occurs that forms a new valley, or the gradual formation of different mountain ranges. These could all operate as potential barriers, even highway systems. Those are huge barriers that fragment populations and different habitats. So again, those are all considerations or types of physical barriers that allow for that separation in allopatric speciation. Now, one thing to kind of keep in the back of your mind is that the actual nature of the geographic separation necessary to isolate populations depends entirely on the biology of that organism, as well as potential or mode for and of dispersal. So it's not kind of this uniform approach, right? It's not uniform or general in nature. If you think about it, if you have the formation of a lake system, right, that separates a population of flying insects, well, chances are those individuals from each population can still fly back and forth between the populations. So you would still have the continuance of gene flow, and therefore speciation wouldn't occur. But things would be very different if we were talking about a rodent population that was suddenly separated. If you had two rodent populations that became divided by the formation of that lake, it's likely that you would not have gene flow. Gene flow would be highly improbable because your rodents can't swim across the river, they can't fly across the river, and therefore speciation would be more probable and would likely occur in that situation. So when you talk about the potential for allopatric speciation, you have to consider the biology of the organism that you're talking about, or the organism or a set of organisms of interest, and their mode or potential for dispersal. So let's look at an example of this and kind of go through it step by step. Again, when we have allopatric speciation, we're starting with a parent population, some physical barrier forms, and then you reach reproductive isolation. Once you reach reproductive isolation, you have the formation of new species. But some people don't really consider kind of the nitty-gritties that occur in the process, and sometimes they'll have a failed perspective of the concept, which is why I think it's necessary to take the time to walk kind of through the steps of the mechanism. So you start again with this parent population. Here our parent population is a happy population of wild horses. They are all able to kind of interact and kind of move or mate amongst themselves freely, right? So you have this free flow of genetic material. The population itself has a common gene pool with regular gene flow. Again, that means each individual has potential access to all members of the opposite sex for mating purposes. And maybe the environmental conditions happen at this time to be very favorable for the species. So it's able to kind of expand its territory a bit to occupy a larger home range and different environments. But over time, you have this gradual formation of a physical barrier. And we're actually looking at two physical barriers that come to separate this once kind of homogeneous population and acts to hinder gene flow between these now isolated populations. Right, so we have the formation of a river here that separates kind of these individuals at the periphery to form isolated population B. We have isolated population A. Population A is isolated from population C because of the formation of this mountain range here. Right, so each of these populations originally was one entire population. Now it's been subdivided into three individual populations because of these geographical barriers. Right, so this is the geographic isolation stage, right? The formation of those new isolated populations. Now, those isolated populations may be or will be subjected to different selective pressures. And those pressures will favor those individuals with traits that are better suited to each environment, right? Based on the course of natural selection, what we know of kind of the operations behind natural selection. So allele frequencies for certain genes will change, and the populations begin to change. And initially, they take on kind of the status of subspecies, right? So subspecies refers to the fact that these populations have undergone different evolutionary changes, but they haven't been diverged from one another long enough to have the establishment of reproductive isolation. As I was saying earlier, in order to truly have the formation of distinct new species, they need to be separated from one another for an extended amount of time, a certain period of time that enables for the accumulation of differences that make them incompatible with one another. That truly prevents kind of these inbreeding or interbreeding events from occurring. Once that happens, you have reproductive isolation. So before you can have reproductive isolation set in, you have subspecies. Now, at this point, these subspecies will continue to undergo changes. So each separated subspecies will undergo changes in its genetic makeup, in its behavior, etc. And this, over time, will accumulate as differences between the groups and prevent mating with individuals of the other populations. Right? So each, at that point, each subspecies gene pool becomes reproductively isolated from the others, and then they attain species status. So before you can have reproductive isolation, you have the formation of subspecies. If they are separated from one another for an appropriate amount of time, that you have that kind of complete evolution or complete change in the genetic makeup and behavior that acts to prevent mating, that's when you attain reproductive isolation and therefore species status. So that kind of has to happen first. It's not as soon as these guys are isolated, you have reproductive isolation. It's a slow process. Again, macroevolution takes large amounts of time over evolutionary history. So don't make that mistake thinking that it's once you have that barrier, suddenly you have brand new species. Right? This is how we have the distinction between new species and subspecies. Right? Subspecies can still, they're different, but they can still interbreed with individuals of the other populations that they once were separated from. So you need kind of that element of time and appropriate change before you can have the true status or distinction of new species. So at this point, you can kind of say that there's two potential results of allopatric speciation. You can either have what are called allopatry or sympatry as far as types of species. In both cases, you have the creation of new species because of this barrier. Most oftentimes that barrier or that physical barrier will remain. So you have allopatric species, right? These are closely related species that are still geographically separated. But you can also have the removal of that barrier for a variety of reasons. But let's say that barrier, that geographical barrier, is removed. And now these closely related species can overlap, right? So they can now have an overlapping distribution. We would call these sympatric species, right? They once were truly geographically separated from one another. Now their populations overlap. Even if the geographical barriers are removed to allow the mixing of the populations and overlapping of their distributions, they will not interbreed because genetic isolation is complete. Right? Once you obtain the status of reproductive isolation and the status of species, these two entities are kind of genetically distinct from one another, which makes them incapable of interbreeding with one another. And we'll talk about kind of the reproductive isolating mechanisms towards the end of this lecture. But again, even if that barrier is removed to allow for kind of the formation of sympatric species arising from originally allopatric species, they will not interbreed. Even if they're overlapping one another, they will not interbreed. Now, kind of another consideration with allopatric speciation, it can kind of be, the process can be separated into two categories, or kind of its operative mechanism grouped by two categories. Those categories being dispersal and vicariance. Dispersal occurs when a few members of a species or a segment of a population migrate to a new geographical area, and a geographical barrier leads to isolation, while vicariance occurs when a barrier naturally arises to physically divide those organisms. Right? So that would be like the formation of a river or a valley. so those are kind of the distinctions between the two. One is, again, caused more by these migrative movements, or the potential for potentiality of dispersion by an organism that then become separated by a geographical barrier. The other deals with the gradual formation of a natural barrier. So again, just two different pathways for allopatric speciation to consider. Now, sorry, so allopatric speciation, again, is kind of the most observable or highly present mode of speciation. This is the frequent one that arises. And we see evidence of these events through a range of different environments across the globe, across a variety of different species. To give you some great examples, we can talk about kind of the allopatric speciation of some species of reef fish. So the formation of the Isthmus of Panama, which occurred about 3.5 million years ago, and currently separates the Caribbean Sea and the Pacific Ocean, that led to the formation of several new aquatic species. You had, again, this formation of a land bridge that now kind of prevented different aquatic species from being able to move freely between those two ocean spans. Now the again, that landmass, it originally formed because of movements in the tectonic plate. So you have these movements in the tectonic plate that gradually created this land bridge or landmass that then acted to block gene flow between populations of different marine organisms. And one of the species that was affected was a species of porkfish. And the arisal of this land bridge separated that species into two separate species. One that inhabits only or is confined solely to the Pacific Ocean. The other species of porkfish that is isolated to the Caribbean. So again, the formation of these two species was spurred by the formation of this land bridge, the Isthmus of Panama. Once that came into play, they could no longer have the free-form movement of gene flow between that original species. Another example is quite close to home here in the U.S. Deals with some of the ground squirrel species and populations in the Grand Canyon. Oh, Figaro, do you want to come up here? Come here. Come here. Don't bring your squeaky toy. No, you can come up here, not the squeaky toy. So the Grand Canyon itself was carved out very gradually. Again, all of these processes are very gradual by the Colorado River over millions of years ago, and the process itself took millions of years. But before it formed, there was only kind of one species of squirrel that inhabited this area. More specifically, they inhabited the ponderosa pine forest that was around the rim of the Grand Canyon. And these were, this single species was known as the Albert's squirrels. About 10,000 years ago, when that we had the ending of the last ice age, the canyon was shaped and changed in a way. So you had changes in the physical formations of the canyon that hindered and then came to prevent the movement of the different populations of that single squirrel species. The canyon itself became, as it got deeper and deeper, and as that Colorado River that was forming that canyon, as that became a larger river, both of them became greater barriers, to the point that the squirrels could not move across those barriers. So they became isolated on either side of the Grand Canyon. So you actually have this physical separation kind of that occurred between the forest and that squirrel species, so that you have a different squirrel population along the north rim and a different one on the south rim. So because these squirrels on the north and south sides were reproductively isolated from one another due to that deep canyon formation and the inability to cross the Colorado River, they eventually diverged into two different species. So now we have the kind of the Alberts, or what we recognize as the Alberts squirrel, and the Kayob squirrels. There was also another species of squirrels that was also kind of divided or isolated to form two distinct species, one inhibiting the south rim, one inhibiting the north rim. But this is, again, a neat example of kind of observing firsthand allopatric speciation in mammal species. Now, even if we changed kind of the stance of this environment, if somehow we removed these two geographic barriers, these species would not be able to interbreed, right? Because their alleles have gone through so many changes that they aren't compatible for mating. They can't mate with one another. So again, they're an example of allopatric speciation, and kind of their inability to mate with one another is true recognition of allopatric speciation and the fact that these are different species. All right, so enough of allopatric speciation. Let's move into the other forms or modes of speciation. So we'll move into sympatric speciation. Sympatric speciation occurs when two individual populations diverge from an ancestral species without being separated geographically. So speciation occurs even though you have two groups that are living in the same area as one another. And even though they are living together and kind of have overlapping home ranges, these organisms from the original ancestral species become reproductively isolated, and that divergence occurs without a physical separation. You can remember that kind of mechanism based on the name. So the prefix sym means same. So sympatric means same homeland. In contrast, allopatric means other homeland. Right? So again, you have reproductive isolation of individuals or a group that have the same home range as the other population, right? So both populations overlap, but they become reproductively isolated. Now, at first, the idea might seem a little weird. Like, how does that happen? Especially after thinking about allopatric speciation. Why would a group of organisms in a population suddenly stop interbreeding when they still live in the same place? And it's a good question. There are several ways that sympatric speciation can happen and how it can operate. There's one mechanism in particular that's most common, particularly in plants, that involves chromosome separation errors during cell division. And this creates genetic polymorphism. Right, so we're going to take a closer look at that process. So sympatric speciation itself can occur when one individual develops an abnormal number of chromosomes, either an extra set of chromosomes or fewer chromosomes. And by such, viable interbreeding can no longer occur between individuals. Now, this speciation phenomena most commonly occurs through something called polyploidy. Polyploidy is a condition in which a cell or an organism has an extra set or sets of chromosomes. To be more specific, an offspring or a group of offspring will actually be produced with twice the normal number of chromosomes. And again, that abnormal number of chromosomes results from an error in meiosis, wherein all of the chromosomes move into one cell instead of separating. So during metaphase, when you have the lining up of the homologous pairs of chromosomes, and then moving into anaphase when you have the separation of those homologous pairs. Instead of moving to separate poles so that they both go to opposite sides and you have the equal dissemination between what will become two daughter cells when we're talking about your first round of meiosis, they all go to one cell. So one cell gets nothing, the other gets all of them. So that's how you get these abnormal number of chromosomes. Scientists have identified two main types of polyploidy that can lead to reproductive isolation of individuals in a polyploid state. Again, when they're in a polyploid state, they are unable to interbreed with normal, quote unquote, normal individuals. In some cases, a polyploid individual will have two or more complete sets of chromosomes from its own species, and that's a condition called autoploidy. That occurs because their own gametes don't undergo cytokinesis after meiosis. On the other hand, when individuals of two different species reproduce and form a viable offspring, such that the extra chromosomes come from two different species, the result is an allopolyploid. Right? So a polyploid itself is either an autopolyploid or an allopolyploid, depending on where these extra sets of chromosomes are coming from. Again, if those two or more complete sets of chromosomes are from its own species, that's autopolyploidy. So it's multiple chromosomes from itself, from one's own species. The other polyploid state is the allopolyploid. So allopolyploid is when that polyploid receives an extra set of chromosomes from two different species, right? Because two different species came together to reproduce or form that viable offspring. Now, once a species develops an abnormal number of chromosomes, it can then only interbreed with members of the population that have the kind of the abnormal number as well, which from that you can have the development of a new species. This again is what we see oftentimes with plants, because they either have to interbreed with members of that same population with an abnormal number, or they cannot interbreed at all. But with plants, they can either kind of circumnavigate this issue where the gametes would be incompatible with normal gametes to produce kind of your normal plant species. The reason they can circumnavigate that is because plants can either self-pollinate or they can reproduce with other autopolyploid plants with gametes that have that same diploid number. So in this case, sympatric speciation can occur very quickly by forming offspring that are called tetraploid, or offspring that have quote-unquote 4n. So again, they have twice the normal number of chromosomes as a normal diploid offspring should have. Now these individuals would immediately be able to reproduce, again, only with those of this new kind, or ones that have a similar number. They would not be able to reproduce with those of the ancestral species. So again, that's how you have the development of this new species due to kind of the development of these abnormal chromosome numbers. All right, so again, that's kind of one mechanism or example of how sympatric speciation can occur. Can sympatric speciation occur without polyploidy? So can it occur by some other mechanism? Absolutely. There are some very interesting examples where it seems that you have sympatric speciation occurring when subgroups in a population use different habitats or access or utilize different resources, have different behavioral preferences, or changes or differences in temporal activity. These can all or are proposed and have kind of been observed to serve as potential mechanisms for allowing sympatric speciation to occur. Again, where you have reproductive isolation of a population within the original species. A really fascinating example of this is with indigo bird species. Indigo birds are brood parasites, meaning the female, when she lays her eggs, she will lay an egg in another songbird's nest. Right? She will essentially choose a host, that host being different songbird and the nest of that songbird. She'll lay her egg in that songbird's nest and then leave. So that songbird who owns that nest is basically burdened with taking care of that bird's offspring when it hatches. And usually the indigo birds, the offspring are much larger in size than the young of the songbird, so they can actually kick the young out and have higher success. So their nest parasites, they hijack the nest of a different bird species and lays their eggs there, and then that original bird species whose nest that is has to take care of the young. But the indigo bird species itself have some really fascinating behavioral mechanisms as far as song learning and sexual imprinting. So male indigo birds kind of have this imprinting on host vocalizations, where they will learn to mimic the song of the host or the song of the bird that whose nest that they were born in. So they will kind of imprint on that host song and develop that in kind of their repertoire that is formed by these species-specific vocalizations that again are learned from the host and then can be learned by other male indigo birds that are mimicking the same host. So the males again take on these kind of a vocal imprint as far as the host and have that example of host specificity. Females will use these song cues to choose both their mates and the nests they parasitize. So the female preferences for both the male song and the host nest results from her imprinting on the host. So when she's born in this host nest, she imprints on that host. So she again selects for mates that mimic that same host, and she will lay or selects for host nests that are of the same host. So when she goes to lay her eggs, she's looking for kind of the same host that she was raised by. Again, this is behavioral imprinting. Now, every so often, you get kind of this misimprinting per se. Rarely the females will lay in the nest of a host that's different from the host that she imprinted on. And she rarely will lay in the nest of what we call a novel or alternate host, but sometimes it happens. And in that case, you get essentially this hybrid indigo bird, right? The resulting offspring, if laid in a nest that's different from the host of mama bird, right? So if mom does lay in the nest of a novel or alternate host, the resulting offspring will imprint on that novel host. Again, that novel host being different from the host of mom bird, and therefore that offspring will become reproductively isolated from the parent population, because that offspring, whether it's male or female, will imprint on this novel host. So it will learn or kind of imprint on the behaviors of that host. It will learn or mimic the host song of that new host and include the different songs that are unique to that host species in its repertoire. And if it's a female, it will prefer males that are of kind of that same host or mimicking that same host, and we'll choose that host to kind of serve as a nest site again in the future. So when you have that misimprinting, when mom lays in a nest that is not of the same host that she imprinted on, you can have what we would refer to as these hybridized offspring. The offspring is different from their parent population. And it's to the degree with some of these new kind of subspecies or species of indigo birds that you will see kind of this reproductive isolation as a consequence of the differences or kind of quote unquote mistakes in host colonization. But you'll also see kind of the divergence of other character traits. So they will begin to exhibit kind of host-specific mimicry of different colorations or markings, and you'll see this actually happening very quickly on the indigo bird chicks, essentially within over the course of one generation. So very quick kind of evolutionary turnover. But it's a fascinating example of how you can have kind of the formation of these new species within a subsequent generation because of behaviors, because of their behaviors of imprinting on the songs of their hosts. And again, when you have a chick that's laid in the nest of an alternate host from mom, that's where you see the rise of the sympatric speciation, where you start getting indigo birds that are different from that parent population. They have different song repertoire, different mate preferences and choices, different preferences for nest sites, and even different morphological characteristics. So this is a fascinating example, and if it really gets you as excited as it gets me, I suggest you read more about it, because this is an area that we're gaining more and more insight on, and it's one that I think can really spark a lot of fascination as far as how quickly some of these speciation events occur, and how subtle the reasonings behind it can be. Another example is with the apple maggot fly, more specifically the North American apple maggot fly. As the name suggests, this is a species that feeds and mates on apple trees. Now, this wasn't always the original kind of host or behavior per se. The original host plant of these flies was actually hawthorn trees. It was only when European settlers first introduced apple trees about 200 years ago that some flies in the population started to exploit apples as a food source. food source instead. Again, these apple trees were brought in, represented a new niche that was open and available, and some individuals exploited that available niche. And from that, you had kind of the start of this sympatric speciation between populations. Flies that are born and were born in apple trees tend to feed on the apples and mate with flies that only feed on or kind of live in those apple trees. Flies that are born on hawthorn trees tended to, or usually will stick with other hawthorn flies. So they kind of prefer to mate with individuals that arise from the same tree type as them. And in this manner, the population over time was effectively divided into two groups with a limited gene flow between them. Even though there was technically no reason why an apple fly couldn't fly over to a hawthorn tree or vice versa, but it seemed that they had behavioral preferences that prevented such action, or even technically you could say that the desire for completing such. So over time, these populations diverged into two genetically distinct groups with distinct different adaptations that were specific for kind of the host fruit, right? So there were adaptations and genetics changes that were specific for those maggot flies that were feeding on apple trees. And those were independent and different of those feeding on the hawthorn trees or hawthorn fruits. And here we started seeing these kind of temporal changes. So the two flies emerge at different times of the year. And this emergence is genetically signified difference that essentially synchronizes them with the emergence date of the fruit of the tree that they live on. So again, that's temporal isolation because they emerge at different times of the year. They don't interbreed. Also based on some of those behavioral preferences that they exhibit, they don't interbreed. So that's, again, another example where you see this non-physical mechanism acting as an isolative mechanism. All right, so that is sympatric speciation. Again, speciation occurring because you have the reproductive isolation of different populations by non-physical barriers. The last type of speciation that I'll talk about is parapatric speciation. Parapatric speciation is very interesting. It's difficult for some people to wrap their head around. It's also extremely rare. This type of speciation occurs when populations are, again, not separated by a geographical barrier, but rather there is some extreme change that occurs. So usually this is an extreme change in habitat, and this causes a smaller part of the population to become isolated. Usually that group is at the periphery of the larger group, and they become differentiated to the point of becoming a new species. Sorry, I have a frog in my throat again. And at this point, we say there's kind of this event known as divergence across a cline. A cline is a gradient of variation in genotype and phenotype. So again, here you have this original population. There's some sort of change that occurs that causes the kind of individuals that are at the periphery of the original population to start undergoing genetic changes. So across this area, you see, again, this gradient of variation in genotype and phenotype between those populations. So parapatric is interesting, not only because you have kind of this weird process through which species are formed, but also because these adjacent populations, even though they evolve into distinct species, they do maintain contact along their borders. And because of that, there is potential for this free exchange of genes between the two populations of the organisms, because they live directly adjacent, but again, they occupy environmentally different habitats. So because they're adjacent to one another, if that barrier that's formed kind of creates this incomplete reproductive barrier, you can have gene flow remain possible between those two populations. And this forms hybrid zones. So hybrid zones can be observed at the interface between these two populations. Hybrid zone specifically is a region in which members of different species mate and produce hybrids. Hybrids are the offspring of, or the offspring products when you have mating events between individuals of two different species. Right, they're a little bit of both species, hence why they're called hybrids. Now, although the individuals from the two populations can interbreed, the offspring will not necessarily be successful in either habitat. So natural selection can operate to kind of promote mechanisms that kind of decrease the number of crosses between such populations. Usually you'll see hybrids having a reduced fitness compared to the parent species, or they will be fertile and thus have no genetic viability. So usually you can, sometimes you'll, if you have these hybridization events, you'll have the creation of hybrids. You don't necessarily have the continuance of a hybrid population as far as reproductive functioning hybrids. Sometimes you do. Most times the hybrids are unsuccessful. But it's an interesting concept, and again, you see across these populations these variations or gradual variations in phenotype and genotype. One of the best examples of this type of speciation is with ring species. Ring species are a distinct type of cline where the geographic distribution of the species takes on this kind of circular appearance. So it's circular in shape, and the two ends of it are kind of have these two adjacent populations that rarely interbreed. But the populations elsewhere along that cline interbreed. So it creates really interesting issues when you're trying to group these individuals into discrete kind of species categories, because at the opposite ends of the spectrum you don't have interbreeding, but along the internal aspect of that cline you do have interbreeding. So you do have the creation of hybrid populations. So again, very interesting example. All right, so that's kind of finishes up parapatric speciation and the major modes of speciation. Now, as I said in the previous lectures, when we talk about species in general, kind of with that superficial definition, we look at or kind of define organisms as being of the same species based on whether they can interbreed to make fertile offspring. And when we talk about kind of that distinction, again, these separate species are reproductively isolated such that you can't have hybridization between the two species. But what exactly is it that causes different species to not be able to interbreed, or why are these species unsuccessful oftentimes when they interbreed to create hybrids? It's an interesting question, and broadly speaking, different species are unable to interbreed and produce healthy, fertile offspring because there are barriers that are set in place called the mechanisms of reproductive isolation. Isolating mechanisms prevent, again, interbreeding and serve to maintain reproductive isolation between different species. And these mechanisms themselves can be divided into two different types based on when they act or occur. There are prezygotic and postzygotic mechanisms. So mechanisms that occur before fertilization are called prezygotic isolating mechanisms. Isolating mechanisms that occur after fertilization are postzygotic isolating mechanisms. So we're going to go through some of the different types and how specifically they serve to maintain reproductive isolation. Starting with prezygotic barriers or your prezygotic mechanisms. These are isolating mechanisms that occur, again, before fertilization. So these barriers prevent members of different species from mating to produce a zygote, or that single-celled embryo. Right? So it prevents the creation or formation of that hybrid zygote. And there's several types or subsets of this barrier. In other words, modes at which this barrier can kind of enforce that ultimate goal, per se. Two species might have differences in habitat preference. And that would arise or cause habitat isolation. So if two species occupy or prefer different habitats, it's unlikely that they would ever encounter one another. So based on those preferences, they are isolated from one another. Great examples with your garter snakes. Garter snakes occupy different habitats. You have water-dwelling garter snakes and terrestrial garter snakes, and they're different species because they're isolated from one another based on the habitat or the preference of the habitat that they inhabit. You can also have temporal isolation. So I had mentioned temporal isolation when I was talking about the apple maggot flies, and that interbreeding between the maggot flies of kind of specific to the apple trees and that species specific to the hawthorn trees is prevented because they emerge at different times during the year. Right? Their emergence cycles are genetically synchronized to kind of the fruiting time or production time of the tree that they prefer, meaning the tree that they feed on or mate on, live in. That's temporal isolation. Temporal isolation deals with or causes two species to become isolated because they reproduce or breed at different times. You can think of it as meaning isolated in time. So because they breed at different times, they are prevented from meeting up kind of with one another and interchanging between mates. Now these, when we say different times, these can be differences in the time of day, it could be different seasons, maybe even different years. But because of that difference at the timing at which breeding or mating events occur, you have the isolation of interactions between those species. Another example, so going away from our flies, is with these adorable little fuzz bugs. These are the eastern and western skunks. They are both spotted skunks, but they are two different species of spotted skunk because they have different breeding times. So because they have different breeding times, they cannot interbreed with one another. That keeps them reproductively isolated and prevents them from ever mating with one another. So that's another form of or example of temporal isolation. Another type of prezygotic barrier would be behavioral isolation. So behavioral isolation goes back to our indigo birds, where you have kind of mate choice based on certain preferences that stem from behavior. Right? With the indigo birds, the female chooses or prefers mates that mimic the song of the same host that she's imprinted on, and also selects her nest based on kind of host imprinting. So that's a type of behavioral isolation, right? So different behavioral patterns or preferences serve to isolate these populations. They could have unique behavioral patterns or courtship rituals that isolate species. Different vocalizations can also serve as a kind of a isolating mechanism as far as behavior, because the members of that species will identify similar members and attract mates of the same species based on those behaviors. So an example that's given here is the blue-footed boobies. Blue-footed boobies mate only after a courtship display that's unique to their species. So the female will only mate with a male that performs that unique courtship ritual. Another type of barrier, so there's mechanical barriers. This is when there's a physical or biological difference in structure that prevents mating. So their reproductive structures are physically or biologically different. They might have different sized bodies, or the reproductive structures simply don't match up together. The example given here is with two types of snails, which are separate species. And we know they're separate species because they have opposite spiral patterns in their shells. And because of that, they have opposite placement of their kind of reproductive organs and sites for mating, such that they don't align with one another. So they physically can't mate with one another because their structures, their reproductive structures, don't work. They're incompatible in that way. The last type is gametic isolation. So this will occur because two species produce egg or sperm cells that can't combine in fertilization, even if they meet up through mating. So you can have two individuals from different species copulate, but because of either biochemical differences that present the sperm from one species getting into the protective barrier of the other animal's ova, or the sperm being incompatible with the egg of another species, or incompatible with the reproductive tract of that species, they are not able to fertilize. So this prevents fertilization even if copulation occurs. And a good example of this is with your sea urchins. That's what differentiates between red sea urchins and purple sea urchins, is that their the sperm of one species is incompatible with the ova of the other. So those are prezygotic barriers. Again, they function to before fertilization to prevent fertilization from occurring between the sex cells of individuals from two different species. Now with post-zygotic barriers, things are a little bit different. So these are isolating mechanisms that occur after fertilization. So two individuals from different species are able to copulate, and fertilization does occur, which means you do have production of a hybrid zygote. These mechanisms occur at that point to deal with this hybrid zygote that has now been produced. A lot of them operate to prevent this hybrid zygote from developing to a healthy, fertile adult. A lot of them are kind of natural in origin, in that they're related to the hybrid embryo's kind of mixed set of chromosomes, which may not match up correctly or carry a complete set of information. In some cases, the chromosomal mismatch is lethal to the embryo, or results in an individual that can survive but is extremely unhealthy, has very low body condition and therefore poor survivability following later in life. Again, that can arise if you have the genes of different parent species interacting in ways that impair development of the hybrid. This is kind of impairing hybrid viability. So right now we're talking specifically about a type of postzygotic barrier called reduced hybrid viability. Two really great examples of this. One is with ligers, right? So there was this big thing several years ago where people were like, oh my gosh, let's make ligers. So they bred a lion and a tiger together, produced this hybrid offspring called a liger. Ligers generally don't survive into adulthood because they physically cannot support their body size. Their heart is too small to support the enormous body size that's created based on the mismatch of the genes they receive from the two different species. So they end up developing very poor body condition. They have very poor cardiac function and respiratory function, and eventually die and essentially suffocate because the heart essentially wears out. The heart is not able to support that body size, which is extremely upsetting, and why people should not do stupid things like that. There are reasons, again, why species don't interbreed, and that's one of them. Another example is with this salamander genus, Ensatina. This specific species of the genus can interbreed and reproduce to create hybrid offspring, but the hybrids do not complete development, and those that do, so sometimes they can complete development. If they do, they're very frail and underdeveloped, so they have low survivability. So that's kind of one way of how the hybrid zygote is dealt with after fertilization. So again, that's one of your postzygotic barriers. Another one is reduced hybrid fertility. So in some cases, a hybrid can survive well into adulthood, very fat and happy, but is infertile because it can't split its mismatched chromosomes evenly into egg and sperm. And that produces a hybrid or a sterile hybrid. And that's this mechanism here, hybrid sterility or reduced hybrid fertility. All right, so example of this is with your mules. So mules, when you have a female horse and a male donkey mate, they produce hybrid offspring called mules. And although mules, like this little guy here, they're perfectly healthy and they can live to ripe old age, but they are infertile. They cannot have their own offspring. And that's, again, because of the inability to split those mismatched chromosomes to form gametes of sex cells. So because of this, going back to the consideration that to be a single species, based largely on the biological species concept, a group of organisms must produce healthy, fertile offspring when they interbreed. Based on that notion, that's why we consider horses and donkeys to be separate species, and why mules are just recognized as the hybrids. They don't have a true species recognition. The other mechanism type of your post-zygotic barrier is hybrid breakdown. So hybrid breakdown refers to the fact that the hybrids themselves can reproduce, but their offspring cannot. So the hybrids are fertile and viable in the first generation, but when they mate, the offspring they produce is sterile. You see this in many strains of cultivated rice. So that's hybrid breakdown. The first generation is fertile, but that's second generation that's produced from the hybrids is sterile, it's non-fertile. So these again are your barriers that operate to keep species distinct. They also function in allowing for the continuation of new species being formed. Remember, it's the whole concept of having a new species truly form is on the basis that it becomes reproductively isolated. So by having these mechanisms, you can maintain or obtain reproductive isolation. So that wraps it up for this lecture. I know it was pretty long in duration, but I hope you got a lot of information out of it. I know that there was some information that I kind of reiterated a lot, but I think by having you constantly return to familiar information, you better learn it. It becomes more recognizable, and it's more likely to stay in your long-term memory rather than short-term memory, which is good for you guys when tests come, which happens to be next week, so please make sure you're studying this weekend. But if you're not in my class, I hope that whatever reason you were watching this video, this video helped kind of satisfy that reasoning, and I hope you're able to walk away with a better understanding of the information, learning it in a better light than you previously saw it in, and also able to take away something you didn't know before.